This video explains LPS. For information on the evidence supporting LPS, please watch the separate video provided. LPS is a component of gram-negative bacteria. The photo in the center shows a bacterial colony, viewed under an electron microscope. A lipid bilayer membrane can be seen in the enlarged image of a single bacterial cell wall, with LPS embedded in the outermost layer. Structurally, LPS is composed of lipids and sugars. Bacterial-derived LPS is found in edible plants, because bacteria live in the soil and on plants. This table shows the LPS content in edible plants and herbal medicines that are considered to be beneficial for health. LPS is consistently detected in these edible plants. LPS is a component that we unknowingly consume through our daily diet, therefore, we all must have ingested it. Herbal medicines, in particular, contain high concentrations of LPS. Therefore, we believe that LPS contributes to some of the effects of Chinese herbal medicines. Here, we will explain about mechanism of action of LPS. LPS acts on cells by binding to a receptor, known as TLR4 on the cells. LPS bound to TLR4 triggers a complex signaling process within the cell, resulting in the perturbation of genes in the nucleus. The elucidation of this signaling mechanism was awarded the Nobel Prize. TLR4 receptors are present on a variety of cells. In particular, immune cells express many TLR4 receptors, which means that these cells are strongly activated by LPS. The photo shows rabbit alveolar macrophages stimulated with LPS, in which morphological changes are observed. Now, let us clarify a bit about macrophages. Macrophages have a special function of ingesting and processing foreign substances in the body, through a process known as phagocytosis. Foreign substances in the body include pathogenic bacteria and viruses that enter from outside, as well as dead cells and cancer cells. Macrophages process these substances through ingestion. The photo shows macrophages surrounding and phagocytizing a dead white blood cell. Thus, macrophages contribute to our health in many ways through phagocytosis, such as defense against infections, waste removal, wound healing, and metabolic regulation. LPS, through its activation of macrophages, contributes to our health. Not only LPS, but also other microorganisms' materials, can activate macrophage. For example, beta-glucan from mushrooms and yeast, as well as peptidoglycan from lactic acid bacteria, also activate macrophages. The vertical axis of this graph represents the level of macrophage activation, based on nitric oxide production. All of these substances activate macrophages. However, compared to beta-glucan and peptidoglycan, which exhibit activity in the order of microgram, LPS exhibits equivalent or greater activity in the order of nanogram. This means that only 1/1000th of the amount of LPS is needed. These results suggest that LPS can be a superior macrophage activator, compared to beta-glucan and peptidoglycan. It is important to note that LPS taken orally is not absorbed into the body. LPS taken orally activates immune cells in the mucous membranes of the oral cavity and intestines. The abilities to produce antibacterial substances and to phagocytize foreign substances are increased in mucosal cells, stimulated by LPS. In addition, mucosal cells stimulated by LPS transmit activation signals throughout the body, through the secretion of liquid factors and migration. The effects of LPS have been verified at the cellular level, in animal experiments, and in human clinical trials. LPS has preventive and ameliorative effects on various diseases, including infectious diseases, metabolic syndrome, allergic diseases, and Alzheimer's disease. The details are explained in a separate video. Thank you for staying with us until the end. For information on the evidence supporting LPS as a food ingredient and as a cosmetic, please watch the separate video we have provided.